Yeah, okay, I've started the recording.
Perfect. So welcome everybody. It's my great pleasure to introduce Vanessa today. Vanessa Sancho-Shimizu is one of our leading scientists within the Center for Pediatrics and Child Health, and who is very interested in the molecular basis of inborn errors of immunity. And it's a real pleasure to have you speak to us today, Vanessa. Thank you very much. The floor's yours.
Okay, thank you. I'll say if you have questions, please just jump in, like turn on your mic and stop me. I think that would work better. So please don't hesitate. So thank you for the opportunity to speak, Gareth. I think I just wanted to share a bit the type of work we do, which is on trying to understand life-threatening infections in children by using genetics and really defining inborn errors of immunity, which really gives us insight into the biology. I'm based at St. Mary's. I'm not a clinician, but I work very closely with all my wonderful clinical pediatric colleagues and have a group here. Really, my work is about taking these patients, looking at the genes and trying to identify causative variants. Okay, so so if you take any ubiquitous childhood infection, this would sort of fit the pattern of the disease presentation, where the majority of the population will either have asymptomatic or mild disease, really of no remarkable consequence. And then a small proportion may have sort of mild disease, and it's the real extreme, the extreme rare occasion where you get life-threatening infections. And in this talk, I'll be talking mostly about, sorry, I'm going to put my pointer on, I realize, about HSV1, and specifically within the herpes simplex virus 1 infections, looking at herpes encephalitis as the extreme presentation of this spectrum. I will end with, again, a similar approach we used successfully in defining critical COVID-19 disease as well. But basically, you can take any common childhood pathogen and it would fall within the spectrum, and we really like to focus on that one rare individual to try and understand what went horribly wrong in that individual, because it gives us insight into what normally goes right and what is essentially important in protection against that infection. This is just a rationale for why we're looking at this now. As many of you would already know, actually, back at the turn of the century in the 1800s, childhood mortality was very high. And most of this, a lot of this was due to death by infectious diseases. With the advent of antibiotics and vaccinations from the 1920s and 1940s, we do see this, this number really improve to the point where the impact of childhood infections is much less, yet it's still there. Now, I put this up only to highlight that technically the patients that I'm looking at, which are the patients who present with life-threatening infections, if we investigated them back in this era, we actually wouldn't be able to really do much in terms of the genetics, because these children would often die from infection, and those genes or those variants that may have been causative would die with that individual, meaning that we're unable to actually study that particular variant unless we were able to catch them before the death of the individual. And we really have an opportunity right now with medical intervention. These individuals who would have died back in the 1800s with life-threatening infections can survive, and we can actually understand what the underlying biology was that defined that phenotype. And also this added with the accessibility and accessibility of next generation sequencing, we're able to now actually look at these rare variants and its effect on the population. So really, the time is ripe now to actually look for these type of rare variants that may determine life-threatening outcomes of infection. Very briefly, So there is this genetic theory of infectious diseases. I think I still get quite a bit of pushback, especially from the clinical geneticists, that infection is not a genetic phenotype. And I think if you ask anyone on the street, I think most people probably wouldn't equate infectious disease outcome to genetics, but clearly there is a host genetic component to all this. This is just some history, which I find interesting, just exemplifying the fact that back in the 1800s, there was very high mortality due to infection. Here is Louis Pasteur's pedigree and Charles Darwin's pedigree. And you can see their children there was substantial infant mortality due to fevers. And the idea that host genetics, well, first of all, the microbial theory of infectious diseases was first presented by Louis Pasteur in 1865, which put infectious diseases caused by microbes. And the idea that host genetics can have an impact on this only really took hold with the observation of intrafamilial clinical heterogeneity, as we see here in these two families. These siblings obviously would have shared an environment, yet not all of them, resulted in death. So there is intrafamilial clinical heterogeneity, but even within the population, that you could have clinical heterogeneity to a response to an infection. This then launched sort of the idea that maybe the host can have an impact, and really culminated in the 1950s with the first identifications of the classic primary immunodeficiency genes, XLA or X-linked A gamma-globulinemia genes. I guess then the idea of primary immunodeficiencies really took hold, and that was the main driver of understanding genetics of infection, which then brings us to today. These classic primary immunodeficiencies tended to be these single gene defects affecting multiple opportunistic infections. These are your SCID patients, your XLAs, and your severe congenital neutropenia type patients. But we're also now understanding that there are these more non-conventional PIDs where one gene can predispose you to a particular infection, and just one single infection. And here is a list of a number of these examples. One of the oldest ones is actually meningococcal disease and complement deficiency. There is a very clear link between these two. What's beautiful about these observations is that it underlines the importance of that particular pathway to that infectious pathogen. And not only does it help us understand how to treat the patient and what went wrong in that particular individual, it informs us of what is actually protective against all meningococcal infections. So it is, these so-called experiments of nature we can learn from. And so these are a number of examples. Here are more. I've sort of taken an interest primarily in severe viral infections. We also work on invasive bacterial disease. But here are a list of a number of other single gene mutations identified in various severe manifestations of viral infections. So the list is growing, and really this has exploded over the past 10 years due to next generation sequencing availabilities. Now today I'll be talking primarily about HSV1 and herpes simplex encephalitis causative genes, which we've identified to be defined by toll-like receptor 3 pathway deficiencies. So just a quick note on the update on where we are with the number of single gene defects that we've identified in patients presenting with life-threatening infections, the so-called inborn errors of immunity. Within this phenotype, actually, it's not only susceptibility to infection that is included, but it's also about inappropriate immune response or autoimmune and autoinflammatory phenotypes. But the key being that these are single gene Mendelian mutations that predispose to these clinical outcomes. So the last update was actually in January of this year. There is 431 genes that have been identified to underlie one of these classified as an inborn error of immunity. There is significant locus heterogeneity. So this means that you can have you can have a mutation in the same gene presenting different phenotypes. For example, this is exemplified by STAT1 mutation. So if you can have autosomal recessive STAT1 mutations, which make you susceptible to viral infections and mycobacterial infections. You can have gain-of-function mutations in STAT1 then that predispose you to hyper IgE syndrome. So there is locus heterogeneity basically tells us that there's this, depending on the type of mutation and where it is in the gene, it can have completely different phenotypic outcome. There is also substantial allelic heterogeneity. So you can have, locus heterogeneity is multiple genes contributing to the same phenotype, and allelic heterogeneity is what I just discussed, which is you can have different mutations in the same gene presenting different phenotypes. What the observations of these types of inborn errors of immunity is that you can have susceptibility to a narrow group of pathogens, like I just mentioned. You can have a single gene defect or single pathway that only predisposes you to, say, meningococcal disease. And also, increasingly now, even though these are called inborn errors of immunity, and there was initially probably a bias towards looking at immune-related genes, many of the inborn errors of immunity genes that have been identified recently are extrinsic to the hematopoietic system. And so these are genes that one normally wouldn't link to the immune response, but are involved in the pathway as well. And you can see here, again, the number of inborn errors of immunity identified has really just exponentially increased since the 2000s. So this has all been brought about thanks to sequencing efforts, and there's many ways we can approach this. We can do whole genome or whole exome sequencing. And just to highlight the differences, exome is the expressed coding region of the genome, which represents about 1.5% of the whole genome. The benefit of doing a whole exome is, number one, it's still cheaper to do this. It focuses on the coding region of the genes, which usually is where the low-hanging fruit is. We'd like to see impactful mutations in protein-expressed regions of the gene because it's likely to have an impact on the phenotype. And also, it means you get 99% less data than you would from a whole genome. So it's just easier to handle. Whole genome actually is obviously where we're moving towards. It's still a little bit more expensive than whole exome. I think we'll get to a point where it is going to be the price isn't going to be an issue anymore. However, the data storage is still a little bit of an issue because, like I said, it's much the amount of data for a whole genome per individual is much larger than an exome. The genome obviously covers every single base pair of your DNA, so it includes all the non-coding regions, which classically have not been linked to Mendelian disease, but this is probably due to just the bias of the search of the research. And there are now increasing numbers of non-coding variants that are presenting in a Mendelian fashion, affecting these phenotypes. So increasingly now that whole genome sequences will be available, I'm sure we'll identify many more of those types of sequences. The other thing to note is that actually whole genome sequencing is better at covering the exome than exomes are. So exome, because of the capture method, sometimes you get not very good coverage towards the ends of the exons, and the beauty of the whole genome is that you get much more equal distribution of that. So actually you get a much better coverage of all the coding region in a genome than you would sometimes compared to an exome. So there's pros and cons of using both. A lot of the inborn errors of immunity that have been identified so far has been based on exome results, again, just because of the ease and also the price of the sequencing platform. Just very quick overview of, we talk about sequencing patients and finding a causative variant. It sounds like a single step and we find something and it's very easy. In fact, if anyone has actually done this, you would know that actually there's a huge process of aligning the sequence and quality controlling it, and then trying to identify all the different variants and which one in an average person will find 1,000 new damaging variants, and which one of those is the causative one. I mean, it's not an easy process to go through, but there is a whole variant selection process that we use and seems to work well. But just all to show that it's not a simple procedure, and there is significant input at each level to try and find the actual variant that's causative. So just because we're going to be talking about these examples of herpes simplex encephalitis and COVID-19, and we'll be talking about the different genes and pathways, just a very brief introduction. I'm sure all of you are already aware of the immune response is made up of the innate immune response and the adaptive immune response. And because we're focusing on childhood infections, and in most cases, and at least with the herpes simplex encephalitis, and actually with COVID-19 as well, we are really focusing on the primary response to infection. And hence, because it's that first encounter, it's really the innate immune response that we've focused on in terms of causative phenotypes. And I'll be talking about toll-like receptor 3 deficiency. So this is just a very brief explanation of what toll-like receptors are. I don't know if this is something that you have all already heard of previously, but if I could just very briefly explain it, toll-like receptors represent, these are receptors found in our cells, either expressed at the plasma membrane or in the endosome. And they serve to recognize various molecular patterns or that are found on various pathogens. So basically, they serve as receptors for looking for anything that's foreign and shouldn't be in the cell. And this receptor will then, if it engages with its agonist, will trigger a cascade leading to the expression of genes alerting the pro-inflammatory response, leading to various cytokines, IL-6, IL-1, TNF to be produced, or type 1 interferons. And so each of these receptors, we have 13 in humans, are specifically recognize a specific molecular pattern. And the ones I'm going to focus on today is toll-like receptor 3, which normally is found in the endosome. And actually in certain cell types, in fibroblasts, for example, it is found on the plasma membrane as well. Toll-like receptor 3 recognizes double-stranded RNA. And we know that upon viral infections, there is double-stranded RNA intermediates produced during a viral life cycle in the cell. These are detected by TLR3 and can trigger a signaling cascade leading to the production of type 1 interferons, which then actually induces an antiviral cascade leading to basically the elimination of that viral infection. So I won't go into, there is actually the single gene disorders of toll-like receptors and TLR and IL-1 receptor immunity. We know that these TLRs are determined, they actually signal through various adapters. So TLR3 has a specific adapter TRIF that it signals through. The rest of the TLRs actually signal through another adapter called MYD88. And we know that there are patients who actually have mutations in these adapters, which control all the TLRs except TLR3. And what we found was that these patients actually have a very specific clinical phenotype, which is that they have extreme susceptibility to invasive bacterial disease, specifically invasive pneumococcal disease. And this has been very well characterized. Interestingly, on the other hand, TLR3, and I should note, what is relevant too is a lot of these other TLRs actually are involved in recognition of bacterial pattern associated molecular moieties. So it makes sense that this is involved. Toll-like receptor 3, I told you, is recognizes double-stranded RNA, and actually we identified, this is work done more than 10 years ago when I was doing my postdoc in John Laurent Casanova's lab, when the first few genetic etiologies underlying herpes simplex encephalitis was discovered. And basically, we identified this toll-like receptor 3 signaling pathway to be particularly important for children presenting with herpes encephalitis. So I just wanted to give you that story and how we got there. Like I said, HSV1 is a ubiquitous pathogen amongst adults. Between 60 and 80% of us are seropositive, yet most of us don't remember that first encounter with the virus because it was probably unremarkable. And only in 1 to 2 in a million cases per year do we see children presenting with devastating CNS infection and HSE. And without treatment, as you probably all already know, it's a very high mortality, up to 70% mortality associated with untreated HSE. Obviously, this number has decreased significantly with the advent of acyclovir, and although mortality remains low, neurological sequelae still presents as a real problem in these children. And I'd say probably until 15 years ago, this was considered an idiopathic disease and just an unlucky event in a child's in a family where a child presented with HSE. And in John Laurent Casanova's lab, we presented we wanted to understand if there was a genetic cause to this disease. Not only is it rare, we also knew that there was no particularly neurovirulent strain of HSV1 associated with the cases. And an epidemiological survey of French patients with HSE, the childhood HSE patients, there was a very high percentage of consanguinity in those patients presenting with HSE, suggesting possibly a genetic cause. So just to highlight that actually HSE is bimodal in the population, we do see, so between the months of 3 months and 6 years, we see a spike of HSE cases, and we do see another increase after 50. And we believe that these two sort of presentations are slightly determined by different immunological factors. And we focused on the children only because we believe that this is all due to primary infection. Actually, we know in our children, we check that this is due to primary infection. So it is a problem with that first encounter with the virus. So we proposed this hypothesis that HSE patients carry a genetic predisposition to HSE. And to that end, actually within John Laurent Casanova's lab, they decided to recruit patients. And again, because these are very rare, he has successfully now have a global recruitment of HSE patients and has over 300 HSE patients recruited. What is usually asked of the patient is a blood sample, and this used to be a skin biopsy. I think now it's just sequencing, but back in the time when I was a postdoc in 2007, for each patient we asked a blood sample and a skin biopsy. And this will come, the reason why we ask for a skin biopsy will become clear later on. It's relevant to the phenotype that we're trying to observe. From the skin biopsy, we grow out fibroblasts, which are actually a very nice source of DNA, RNA, protein of the patient, and we can do a lot of viral assays using this. Whereas with the blood cells, we could do a functional screen, it's very limited. And the approach back then, because this was pre-next generation sequencing, so please remember that we didn't have access to exome or genome sequencing. This was all Sanger sequencing based on a single, PCR, which was very intensive. We were very much phenotypically driven. So we took the patient's samples and tried to get an informed phenotype from their cells. Basically, we took the patient's blood or skin cells and infected it with HSV1, or took any of the agonists of the toll-like receptor pathway to see if the cytokines were produced. And when we saw an impaired phenotype, then we would focus on that particular patient and then look at the different genes involved in that pathway to figure out what could be the candidate pathway or the gene involved. And actually, the first few genetic mutations identified with HSE was discovered through this route. We also had some consanguineous and multiplex families, which we could do positional cloning on and linkage analysis to inform the phenotype, but actually most of the genes identified were driven through this functional pathway. And through this analysis, actually, then we identified the toll-like receptor single gene mutations on toll-like receptor 3, UNC93B, TRIF, TRAF3, TBK1, IRF3 also now have been identified in children presenting with HSC. And the idea is that HSV1, it is a double-stranded DNA virus. And we do know actually that it can form RNA intermediates during its viral life cycle. And in fact, we believe maybe that's how this triggers the toll-like receptor 3 signaling response. And in the patients with these deficiencies, well, normally, a normal individual would detect the virus and trigger this cascade, leading to the production of type 1 interferons, which then will mount an antiviral program and get rid of the virus. In our patients with these deficiencies, this cascade is blocked, and hence we don't see any production of type 1 interferons. And this really became the basis of the functional assays that we were doing to screen the patients for HSC. And just to give the story of how that first individual, the first mutation was identified. So the first gene linked to HSC was UNC93B. This gene still remains less understood than the other players in that pathway. It is an ER resident protein and absolutely necessary for the proper production of toll-like receptor 3. The reason why this was first identified was really thanks to this paper published by Bruce Butler's group. He was the Nobel Prize laureate for the identification of toll-like receptors. He had carried out this, this came from a mouse study, a beautiful mouse study where he had created ENU mutagenized mice. So he created mutations, single variant lesions within a population of mice, and then looked at its response to different toll-like receptors. And then where he found an aberrant response, he tried to figure out what gene was actually mutated, and that was a way to define a phenotype of a particular gene. UNC93B1 was an open reading frame of unknown consequence until this paper was published. So this mouse, the UNC93B1 mutated mouse called 3B, when they took the mouse cells, what they found is this very peculiar phenotype that the mouse cells were unable to produce or trigger a response to TLR3, 7, 8, or 9. Yet TLR4, 2, 6 were all conserved. And lo and behold, actually, one of the first HSC patients that were investigated in Paris was a child who suffered from herpes simplex encephalitis. I told you we took a blood sample from the patient, and when the functional assay was run using different toll-like receptor ligands, the child was also presenting with this peculiar phenotype of not responding to TLR3, 7, 8, 9. So it was really a wonderful sort of coincidence that this paper came out at the time that HSC patient that was received in Paris had this peculiar phenotypic outcome. And having seen that paper, it was only obvious to sequence the human ortholog of UNC93B, and lo and behold, the patient was found to have a deletion in UNC93B, leading to a defective protein. And there was a second kindred, both of these were consanguineous families, having another homozygous mutation in the same gene. So really just a story to highlight the fact that, human genetics is very important, and these sort of taking advantage of understanding these experiments of nature in patients is extremely powerful. Yet we are very much aided by animal models, and which has really driven our knowledge of and understanding of immunology. So this was a really nice sort of marrying up of those two aspects. So once UNC93B was identified, actually looking at the pathway, we wanted to look to see what other genes in that particular pathway could be involved. And it became very clear that actually TLR3, which is the receptor that UNC93B is actually controlling the expression for, would be important. And so TLR3 was sequenced in a number of HSC patients in the lab, and mutations in TLR3 was identified in a number of HSC patients. So this then sort of begun the identification of genes within that same pathway that may contribute to disease. And because they are on the same pathway, in fact, we can come up with a common response. And again, like I said, the screening method. Now, on blood, actually, looking for TLR3 defects is very difficult. I'll get to it later, but it's only because the agonist or the double-stranded RNA we use to trigger the response can be actually detected by many other receptors in our cells. But interestingly enough, in fibroblasts, TLR3 is expressed on the plasma membrane, making it a very good selective marker for assessing TLR3 function. So this is why we needed skin cells or fibroblasts from the patients to assess the response appropriately. In our patients with UNC93B and TLR3 deficiency, they have this common phenotype. If we take poly-IC, which is a synthetic double-stranded RNA that we sprinkle on the cells of the patients, you see absolutely no interferon beta or lambda, so in type 1 and 3 interferons. And also there's reduced pro-inflammatory cytokines IL-6, which is also controlled by that pathway. Let's see. So this then led to, again, the identification of all those genes that actually fall within that particular pathway, leading to the production of type 1 interferons. So just to highlight what all those genes in that same pathway have in common is they have impaired TLR3 induced interferons. If we infect the cells with HSV1, we see the same impairment of cytokines. If we infect the cells, we also see extremely high HSV1 titers in the patient cells and extremely high cell mortality. We know that if we then take exogenous recombinant interferon and put it on the cells, because this is what is lacking, so it's a lack of interferon that's driving this phenotype, we actually rescue the patient cells. So if we take recombinant interferon, put it on patient cells, we can rescue the high titers, and also we can rescue the high cell death. So just as a sort of a detailed look at how we actually define, how do we go from the actual gene, from the patient to the gene, to proving that the gene or the variant in the gene is causative. I'll just take a very quick sort of example with the gene that I worked on, which is TRIF. So again, it's still in that same pathway of toll-like receptor 3. TRIF is the sole adapter for toll-like receptor 3. So without TRIF, you can't get any signal induction and production of type 1 interferons. So this is just the case history. So, you know, keeping in mind that actually, I think looking at this type of case history, I think most people won't think that this is due to a genetic deficiency. And I think that the point I want to make here, not only specifically with HSE, but with any childhood disease, is any extreme presentation of a common infection should raise flags that the idea that maybe a genetic cause can be at the heart of this. Obviously, there are many things that have to be excluded to get to that point, but any extreme presentation of infection that can't be easily attributed to any other risk factor should be considered as a possible genetic cause. So in the TRIF deficient patient that I worked with, the patient was born to consanguineous parents, but actually this is obviously a big giveaway for genetics. I think any consanguinity is always the first thing you would think is a genetic cause. But most of our patients actually with HSC are not consanguineous, the ones that we have worked on. So this is not, in this case it was, but just because they aren't doesn't mean that it can't be genetic. The family had no infectious disease family history. This is quite typical. The child had viral meningitis of unknown origin at 18 months and then presented again at 2 years. with a very sort of typical HSC picture, was later actually identified to be HSV1 positive in the CSF, so had bona fide herpes simplex encephalitis. He suffered neurological sequelae, actually quite mild neurological sequelae considering, and has negative serology for all other viruses. And so this is also quite remarkable that actually these patients, although they may lack some, an entire pathway that's apparently important for detecting viral infections, they seem to present with a very narrow spectrum of clinical phenotypes. And so with HSC, it really is just herpes encephalitis and no other herpes-related diseases or any other viral infections. I say that now, but I will correct myself at the end with COVID-19, but COVID-19 presents itself as a different scenario. So in any case, this is the case presentation, and I just highlighting the fact that any extreme presentation should be considered, don't rule out genetics. And so in this particular case, a consanguineous patient had presented with HSC. We identified a homozygous mutation, a nonsense mutation in TRIF, which basically abrogated the expression of the gene. So the first thing we do, we sequenced the gene, identified a candidate mutation. This is a nonsense mutation, which is considered quite damaging. We will then, next thing to do is to assess the patient phenotype of that particular gene. So we took fibroblasts from the patient and a control and show that actually patient cells don't produce any TRIF protein. We've also assessed mRNA, it doesn't produce any functional mRNA. And this is the classic phenotype I showed you already, that if we take his skin cells and we trigger the TLR3 pathway, there was no response. So confirming that the pathway is impaired and there is no expression of the TRIF protein, which is necessary for that response to occur. We then infected his cells, again, showing that there is no cytokine production, high cellular mortality, and viral replication, which is rescued upon exogenous interferon alpha. So this is very similar to the other cases I've already showed you. And the one extra step we have to make in proving that this allele is actually causative is to take the patient's cells and basically rescue it by introducing the wild type version of the gene. So in this scenario, we took the patient cells, which on its own, when you put poly-IC, does not respond to cytokines. We took P1 cells, the patient cells, and then we introduced the wild type version of TRIF, and we can restore signaling. If we then take the patient cells and put in the mutated version of the gene that it already carries, we confirm that actually there is no production. So we know that the mutated version that the patient carries is a null allele. It has no function. We were able to do this experiment with the HSV1 replication as well, and confirm the expression of the mutation as well. So it's only really by complementing the patient cells with the wild type version of the gene can we be absolutely sure that this variant is causative. So this is that was just a very detailed look at how we then in the lab take the gene and try to tie it to the pathogenic cause of the viral infection. Taking a step back, just to finish the story and what we've understood about herpes simplex encephalitis, using the knowledge gained from these patients with inborn errors of immunity, we can ask some very interesting questions based on these observations. So we know that these patients presenting with TLR3 interferon-mediated immunity and deficiencies with HSC, they, like I said, they don't have any other problems with other respiratory viruses or any other viruses. And they don't seem to have any other HSV1 related disseminated disease. So, we asked the question, what accounts for the lack of HSV1 dissemination to epithelia or internal organs? We don't see this HSV1 viremia in these patients. This is very typically just encephalitis, so it's only infecting the CNS. Why is that? These patients have a germline mutation in this gene, meaning that every single cell lacks that pathway, yet we have a very tissue type specific presentation. And then what makes TLR3 deficient patients normally resistant to most viruses? As I said, most viruses do produce RNA intermediates. If they can't produce heparin interferons, how is that possible? So to address this, experiments were carried out using patients with TLR3 deficiency. And what's interesting is that actually I showed you in the TLR3 deficient patients' skin cells, we see this deficient interferon response. But actually, if you take peripheral cells, so in the blood, so PBMCs, myeloid dendritic cells, or plasmacytoid dendritic cells, or keratinocytes, and trigger the response with double-stranded RNA, in fact, they have a completely normal response. They don't show a deficiency. And the reason for this is the fact that, like I mentioned, RNA is actually RNA receptors. There are many of them within our cells. So TLR3 is just one of a whole number of other receptors that can lead to the signaling of type 1 interferons. Now, the expression of these various receptors is different in different cell types, and it's really this tissue type determinism that is driving the phenotype that we're looking at. And we know that TLR3 is highly expressed in the CNS, and in the blood cells, TLR3 plays less of a role. And so this accounts for why we don't see any defect in the peripheral aspect of HSC. Now, there was a long-standing problem with the the question that everybody asks at conferences when this was presented was, well, you show that there is a defect on fibroblasts of these patients, but they don't have a skin phenotype. They have a CNS phenotype. How can you prove that, why is that? For a long time, obviously, this was very difficult to do because we had to actually access CNS cells from the patients. But now with the advent of this iPSC technology, or the protocol has really revolutionized patient-derived sort of research. And so iPSCs are induced pluripotent stem cells. Basically, you can take any somatic cell. Mostly it's done from PBMCs or skin cells, fibroblasts. And if you overexpress various, I think it's 4 factors, these Yamanaka factors, you can actually coax this terminally differentiated somatic cell into a pluripotent state. And this pluripotent stem cell can then further be differentiated into various different cell types, which represent, obviously, the patient-derived cells because it's the patient genome. So it's really the question of what happens in these TLR3 deficient patients' CNS cells was only, we could only really answer that once this was came into standard practice. And so this is work done by a very talented postdoc that worked in John Laurent Casanova's lab, who created iPSC cells from the TLR3 and HSC patients and derived neural stem cells, neurons, oligodendrocytes, and astrocytes from those patients. And in that paper, they show that, in fact, patients with any of the TLR3 deficiencies that we've identified actually have a complete lack of control of viral replication in neurons and oligodendrites. Here we have HSV1, which is GFP tagged. This is a culture of neuronal cells. These are control neuronal cells infected with GFP. You can see maybe a few spots here, but control cells are able to control this infection, and also in oligodendrocytes. Whereas in patients with UNC93B deficiency, and I think they also tested TLR3 deficient patients, you see a complete lack of control. So this is the first time they were able to say that this phenotype actually is tissue type specific and has an impact at the CNS level, and it's a very tissue specific observation. So I hope I've taken you through this very detailed example of how TLR3 interferon deficiencies can cause HSC. It's a very narrow defect in antiviral interferons with a very, which causes basically a single viral infection. And just quickly for the last maybe 5 minutes, I wanted to touch on how actually these TLR3 deficiencies have suddenly become interesting, or the type 1 interferon deficiencies have suddenly become interesting in the context of COVID-19 disease. So over the past year, since the pandemic started, we have put together a coalition, a wonderful consortia, actually, again, led by the John Laurent Casanova Group, and also Helen Su at the NIH, called the COVID Human Genetic Effort. And this is, sorry, I don't have a slide, I meant to have one here, is a international consortia of over 80 members, representing basically the global representation. And the idea was to try and tackle the human genetics of COVID-19 disease. And the first question that was asked was, are there any inborn errors of immunity that can determine a critical COVID-19 disease? And so I'm going to talk to you about the results that we got from that. Very briefly, though, I don't think COVID-19 needs much introduction, but the main point being that I think there's lots, it's amazing how much we've learned over the past year, and there's still lots of questions. The main basic understanding of the disease process is that you have this physiological host response that's mounted within the first 2 weeks of infection, which is normal, let's say, and hopefully leads to viral clearance. You get viral entry, replication in the lungs, induction of the host immune response, which is very much driven by type 1 interferon, as well as many other immune modulators. And hopefully in individuals where this, they have asymptomatic or mild disease, this is resolved. And there really is this point, I think usually at 2 weeks, where this transition from mild to severe happens. And suddenly in patients who present with critical COVID-19 disease have a problem with controlling the host response, and they get into this hyperinflammatory phase leading to multi-organ dysfunction. And it's really understanding why or how different patients, how this switch happens, what is going on. And so we again presented, we try to tackle this by asking a genetic question. Just also to highlight that there are many clinical complications of COVID-19, which is absolutely fascinating. And each one of these phenotypes can be seen as an extreme phenotype to be actually further dissected genetically. We focused on critical COVID-19 pneumonia. I'm actually now also working on PIMS-TS, or the multi-inflammatory syndrome in children, as a particular phenotype as well. But I'm just going to talk to you about a critical COVID-19 genetics. And as I said, so to try and identify the monogenic causes of COVID-19 disease, SARS-CoV-2 falls into that same spectrum I presented at the very beginning, where most people are present with asymptomatic or very mild symptomatic disease, and it's only in the extreme scenario do we see this very severe presentation. We also know in terms of risk factors, there are 3 main risk factors. There is the biggest risk factor is age. We know that the older you are, the more likely you are to present with COVID-19, critical COVID-19 disease. There are various comorbidities, and also the sex. So we know that men are at higher risk for this. But at each demographic category, there is an extreme presentation. And this, again, can also be rolled out to other infections. But we try to focus on the young presenting with rare idiopathic severe disease. The other genetic interest is looking at the rare unexplained resistance to infection in the older individuals. So looking at really these extremes is where we would look for genetics. But we focused on, in fact, we were quite lenient in the first round. We took everybody. We didn't take an age cutoff and looked for critical COVID-19 disease across the whole spectrum. So very briefly, 700 patients, 659 patients was included in the study with life-threatening COVID-19 pneumonia. 75% were men, 22% women. This is very, I think, quite classic of the presentation of critical COVID-19 disease. 14% died. And we also had a cohort of asymptomatic or mild cases of COVID-19 to compare to. Now, very different from the approach I took in the HSE case, where we're really looking at sort of personalized genomics and looking at the one individual, one family, and trying to tease it out. We took a cohort approach in this scenario, driven by a hypothesis. that we already knew inborn errors of immunity in type 1 interferons. I've already spoken to you about TLR3. There are others, IRF7, IRF9, and all these other genes in the middle that have been attributed to severe viral infections. In particular, I've already talked about all these genes which predispose to HSC. I didn't mention, but TLR3 deficiency has since been also linked to acute respiratory distress syndrome caused by flu. And this is also found in IRF9 and IRF7 patients. So these patients present with acute respiratory distress syndrome. So there was a hypothesis suggested that do the patients presenting with critical COVID-19 disease have mutations in any one of these genes? We know type 1 interferon is important in the resolution of infection. And so we searched for mono and biallelic non-synonymous variants with that are rare in the life-threatening disease patients, and found 118 variants actually in this pathway. Okay, I didn't include this. So basically, what we identified is there are actually, of those, there's 23 mutations that were actually found to be loss of function and causative. And in fact, that accounts for, I don't have it here, sorry, that accounts for 3.5%, sorry. What we found is 3.5% of all life-threatening COVID-19 patients have damaging mutations in this type 1 interferon signaling pathway. So we know that inborn errors of type 1 interferon immunity are important. What was really nice also is that there was a search for autoantibodies to type 1 interferon. So these are variant antibodies produced to our own protective type 1 interferons. This is very rare in the general population. Actually, we found 0 in the asymptomatic and mild patients. In the patients with critical COVID-19 disease, there were 10% that had these autoantibodies to type 1 interferons, which is absolutely remarkable, and basically neutralized the entire pathway. So what was really nice about these discoveries, there's published sort of back-to-back last September, is that we had a genetic and an immune-mediated cause of critical COVID-19 disease, which highlighted that type 1 interferon immunity was absolutely critical for preventing severe illness in COVID-19. So together, 13.5% of critical COVID-19 patients could be was explained by either genetic mutations in the interferon pathway or because they had antibodies to their own type 1 interferons. So yeah, so that was really quite nice, and actually highlights the importance of type 1 interferon, which we believe is important early in infection. And there was case reports on a TLR3 deficient patient who presented with COVID-19 who was treated with type 1 interferon and actually did not develop severe disease. So, and I think there is also clinical trials underway for this, for COVID-19. So that's it. I just wanted to give you a highlight of the type of work that we do here. Very quickly, just in my lab, we do the genetic basis of severe infections with a particular interest in herpes and simplex encephalitis, but also invasive meningococcal disease. And my research really involves the combination of lab-based science and also clinic. And we have a specific dedicated BRC-funded pediatric infectious disease clinic that Dr. Jethro Herberg actually runs, my very close collaborator, and the rest of the pediatric team at St. Mary's, where we can recruit these patients and we can carry out the research to identify new inborn errors of immunity. And so this is really a true clinical research collaboration with all sorts of clinicians, molecular biologists, computational biologists, and the clinical geneticists. I think Harry actually spoke earlier to you. He's also involved in informing the phenotypes. And that's it. Thank you.
Vanessa, thank you. What an amazing talk, and a huge amount that you've covered there. Maybe should we stop the recording, and if anybody has some questions, we can just check those out.
Absolutely. Let me just.
And I know everybody's really busy and will probably be having to rush off, but has anybody got